Have you ever wondered how we grow up and heal our wounds? Have you ever wondered how we pass our characteristics from one generation to another? The secret lies within our cells, the tiny building blocks that our bodies are made up of. One part of our cells' life cycles is the time when cells divide into two new cells. This is an important stage, since without this, both growing up and passing our characteristics would be impossible. We also wouldn't be able to heal a cut or a bruise. In fact, we wouldn't be able to last a while after fertilization if our cells do not divide. In this video, I would answer how we grow up and how we pass our traits, as well as expound on the magical process of cell division. First of all, before I expound on cell division, I just want to point out some of the parts of the cell that are involved in it. As we all know from previous years, the nucleus is the control center of the cell. It directs cellular operations and contains and transfers the DNA, which is the hereditary material of the cell. Cell division revolves around it. The nucleus contains the nucleolus, which produce protein-producing organelles called ribosomes, and the chromatin, which is a string-like fiber that contains protein molecules in the actual DNA. During cell division, chromatin gets condensed into chromosomes. Around the nucleus is the cytoplasm, a jelly-like layer that contains other organelles like centrosomes, mitochondria, etc. There are two types of cell division, mitosis and meiosis. Mitosis is responsible for growth and healing, produces diploid 2 and chromosome cells, and takes place in regular body cells. Meiosis is responsible for the production of gametes, sperm and egg cells, produces haploid and chromosome cells, and takes place within reproductive organs. Before cells divide, they go through a long interphase to prepare. First, cells go through the gap 1G1 phase, the phase before DNA duplication. Then, most cells go through the synthesis S phase, when DNA is copied, although a few cells enter the G0 phase, where the DNA can't be duplicated. For the cells whose DNA was copied, they enter the gap 2G2 phase, where the cell prepares for splitting. Then, it's cell division time. There are 4 phases of mitosis: prophase, metaphase, anaphase, and telophase. Prophase is when chromatins condense into X-shaped chromosomes. Each chromosome is double-stranded and has 2 sets of sister chromatids. The nuclear membrane and nucleolus disappear and spindle fibers appear. Metaphase is when chromosomes attach to the spindle fibers and align at the middle of the cell. Anaphase is when the chromosomes split into 2 pairs of single-stranded sister chromatids and gets pulled to opposite ends of the cell. Telophase is when the nuclear membrane and nucleolus reappear, spindle fibers disappear, and chromosomes uncoil. The cell cycle culminates with cytokinesis, which produces 2 diploid, independent, and genetically identical daughter cells. On the other hand, there are 2 divisions that make up meiosis, meiosis 1 and 2. Each division has 4 stages: prophase, metaphase, anaphase, and telophase. There are key differences between these stages in mitosis and meiosis. First of all, in prophase 1, the pair of homologous chromosomes pair up, forming tetrads with 4 chromatids. There, the homologous chromosomes exchange some of their DNA. The tetrads align at the middle of the cell on metaphase 1 and split into 2 double-stranded chromosomes in anaphase 1. The result of meiosis 1 is 2 haploid daughter cells. No chromosome replication happens in meiosis 2. Meiosis 2 is much like mitosis. In the end, 4 haploid genetically unidentical daughter cells are formed. Gametes being haploid is actually important since in fertilization, they combine with another haploid gamete and become a diploid cell. If gametes were diploid, then the next generation will have double chromosomes, which would be a huge problem. Aside from cell division, passing of characteristics from one generation to another, in other words, heredity, are also governed by the Mendelian principles of genetics. These are named after Gregor Mendel, an Austrian monk who performed an experiment on breeding garden pea plants. He observed that the peas have traits similar to their parents, however, different traits occasionally arise. He first allowed the garden pea plants to self-pollinate for many generations to create pure-breeding strains. Next, he crossed different purebred flowers from the first parental generation together. For example, he found that all of the flowers from the resulting first filial generation were purple. Finally, he crossed the flowers from that generation together. Most of the resulting flowers were purple, but shockingly, around 1/4 of the flowers were white. After many other contrasting characteristics of garden pea plants were studied, Mendel proposed some laws that are now known as the Mendelian principles of genetics. One of his findings was that genetic characteristics have alternate forms, which we now call alleles. One allele is dominant over the other recessive allele. If one parent passes on a dominant allele while the other passes on a recessive allele, the offspring will express the dominant allele. Only 2 copies of the recessive allele is enough for it to show. But just because an allele is not shown doesn't mean it is lost. Let's go back to the pea plants. Purple flowers are the dominant while white flowers are recessive. The phenotype, or physical manifestation, of the first filial generation was all purple. However, their genotype, or genetic characteristic, was heterozygotic, or that there is one dominant and one recessive allele. This allowed the recessive trait to resurface. This is the law of complete dominance. Another of his findings relates to meiosis. A random allele is passed by each parent to the offspring. Only when the offspring is fertilized do the alleles recombine. This is why offspring have different gene combinations. This is the law of segregation. Lastly, the alleles of different genes don't affect the probability that those genes are passed. This is the law of independent assortment. A popular way to visualize these laws is the Punnett square. It is much like using the FOIL method used in multiplying binomials in math, only this time the arguments are the possible gametes of 2 organisms. The gametes of one organism are placed on top, while the gametes of the other are placed on the left. Then all one has to do is put the gametes in the corresponding row and column together. That way, one can notice the possibilities of the offspring's genotype and phenotype. It even works with 2 traits being inherited at a time. To answer the questions at the start of the video, the reason why we grow up and pass on our characteristics to the next generation lies deep within us, in the cells that make us up. In passing our characteristics, the answer lies even deeper, inside the DNA and genes that makes us who we are. Studies on cell division's relation with growth can be applied when making medicines and chemicals that can cure diseases, even those like cancer. Studies on passing our characteristics can help make people genetically stronger, ethical regulations aside. All in all, big things really are caused by small acts.